Good morning. Again, an announcement. We have the simulcast in 10 Evans. So in the future, if you want to just go straight to 10 Evans, feel free to do that. It holds about 200 students. There's a GSI there. Whenever we do an iClicker question here, we'll collect the data there as well. So we have another GSI there who will be collecting the data. So let's get started. So again, my name is Mike Meehan. Welcome to Biology 1A. You'll also probably be taking the lab class. That's Monday night. Labs and discussions begin next week. Okay, so there are no discussions this week. There's no check-in to lab lockers this week. Everything as far as labs and discussions begin next week. If you're trying to switch either your discussion or lab, please use Telebears. There are some spaces in various labs and sections, so feel free to use that option. Come Monday, you must attend your assigned lab and discussion, all right? GSIs will be taking attendance in discussion, and if you don't show up, we're going to consider that spot available for another student. So any questions about labs and discussions? In terms of this class, there's a course reader, which was currently posted on DSpace, a couple lectures. We'll do that for future lectures as well. If we get a chance for Dr. Dowden's part, she will give the first series of lectures. The second series of lectures will be taught by, hang on a second. Nipa Patel, and the last part by Dr. Firestone. Dr. Patel will probably be posting his lectures on DSpace. It's the first time he's taught. Dr. Firestone will have a reader available. That will be at Copy Central. You'll get more information as we get closer. In terms of textbooks, we are using the 9th edition of Campbell. That's the only one we're referencing. It's been out for quite a few years. We initially were going to use Mastering Biology. We used this last semester. We did a lot of assignments from it, awarded points. A fair number of students thought it was busy work. So we decided not to do Mastering Biology this semester. We will use iClickers during lecture. You will be given points for them. Basically, 1 point for a correct answer, half point if you got it wrong, but you at least participated. So there is that motivation to come to lecture. Now, when we haven't done iClickers in the past, attendance has been anywhere from 30% to 50%, which if clearly you're running a company, you can do it, okay? So with iClickers, attendance is around 95%. Huge difference, okay? So anyway, I had students who never registered iClicker, never came. That's certainly your option, all right? The way we'll work it is we'll try to have about 15 iClicker questions worth 15 points. The maximum you can earn is 12 points. The reason for that is if your iClicker doesn't work someday, or your batteries are dead, or you forgot it, or you missed lecture, you don't have to email us and say, oh, gee, I got hit by a bus or something, okay? We don't need those excuses. We're building into the system the opportunity to miss a few lectures. Is that clear? Likewise, if your batteries are dead, borrow the batteries from another person's iClicker. You can easily take the batteries out and insert new batteries in 15 seconds, okay? So hopefully the batteries will never be an issue. Any questions about iClickers? Okay? We'll do those during lecture. Again, we'll try to do at least one every lecture, and some lectures will clearly have more than one. For those of you standing, it's 50 minutes. We do have space in 10 Evans, so feel free to go there. It will be simulcast there. So iClickers, yes, no Mastering Biology. The 9th edition of the book. Exam dates are listed there for 1A. All of this information is in the syllabus. Please read that carefully. The exams are 8 in the morning on the 25th, the 5th, and the 13th is listed. For the lab class, you'll find out more about this on Monday night. But again, it'll be the 9th edition of the textbook, and those exams are at night on Wednesday, March 20th, and Wednesday, May 1st. Any questions about the exam dates or switching sections or anything like that? All right, we have, I think, about 27 different discussion times in that. We have 9 different GSIs we'll be teaching them. Please go to discussion. I think it's very valuable. In the end, the way grades are given is we'll look at your total points, which will include exam scores, and your iClicker points. We'll make a distribution. Students who are within a point or two of the next higher grade, and there's always about 100 students like that, we consider them to be potential bumps. So when I meet with the GSIs, once I meet with the faculty, we determine the cutoffs. I then tell the GSI the students an A-, but they're close enough to the A, we can consider them for a bump. The people who make that decision are your GSIs. They're the ones who will get to know you better. Please come to office hours. We are really here to help you, okay? So we enjoy it when you come to office hours. I think you'll find it very valuable, but we won't get to know all of you. Your GSIs will get to know you better. That's why they make that decision. And what they'll do is they'll look at, did you come to discussion? Did you participate? Various things like that. Any questions about that bump? No? Okay. Enjoy lecture.
All right. Well, thank you, Mike. That's great. Good morning, everyone. My name is Jennifer Doudna. I'm a professor in molecular and cell biology with a joint appointment in chemistry here at Cal. And I taught Bio 1A for several years in the past. I didn't teach last year because I was on sabbatical, so I'm back now. And it's a pleasure to be working with all of you. So I'm going to be teaching the first 5 weeks of Bio 1A. We're going to talk about molecules, the molecules of life. We're going to talk about the chemistry that underlies the basic biology that you're going to be learning about. And what we'll try to do is to, in lecture, go through the material that I think is the most critical for you to know when you come away from this course. So when you go to the textbook, you'll see that there's lots and lots of information in the textbook. And every year students ask me, do I need to read the book? And do I need to know every single fact that they list in the book? And the answer is, the first question is, yes, you need to read the book. It's very important to read the book. And the reason is that we can't possibly, in 3 50-minute lectures a week, go through all of the material in the kind of depth that would be important for your understanding. So you do need to read the book to support what we're talking about in class, but you do not need to memorize every fact in the textbook. What I'll try to walk you through in lecture is the material that I consider to be the most critical for you to understand. And when you go to study, you should focus on what we talked about in class. You should focus on the summaries that I will give you every probably couple lectures. You'll see in the back of my lecture notes something called "You should now be able to," and there'll be some bullet points. So that kind of is a guideline for what I want you to take away from the class. And you can also go to the problems in the back of the chapters in the book to help you work through the material. And I think I will probably give you 3 problem sets during my section. Ungraded, you don't have to do them, but they will be a good way for you to test yourself to make sure that you're keeping up with the material and that you understand the kinds of questions that I'm likely to ask you on the exam. Okay, now this is a big class, really big class, and I really enjoy teaching this class. I really enjoy interacting with students. I do encourage you to come to my office hours. You can talk to me about the material. We can go through questions that you might have about lecture. We can work on problems together, those sorts of things. We can get to know each other at least a little bit. So I do encourage you to approach me and come to office hours if you can. Also during lecture, if you have a question, please do raise your hand because even though it's a big class, because if you are wondering or something isn't clear to you, it's very likely that it's not clear to a lot of other people in the room as well. So please do feel free to ask me a question if something is really unclear to you during the lecture. I want to start out by actually telling you about a really cool lecture that I hope some of you will go to. It's actually tomorrow night in Stanley Hall, which is just across the way. This is the Regents lecture with Dr. Thomas White. So the title of this lecture is Genomic Medicine, Development, Economic, and Ethical Challenges of Translating Basic Research into Clinical Practice. So a lot of what we do in biological research is discover the fundamental ways that things work in biology. And of course, we always hope, certainly in my own research, I hope that the kinds of discoveries that we're making will have an impact on things like human health. So this is a talk that will directly talk about making that connection. And Dr. White has a lot of experience in this field. He's worked in various companies developing different kinds of technologies. And I think you'll find his lecture very interesting. So I do encourage you to go to that tomorrow evening. And just a couple more practical things. When are my office hours? So I'll be holding office hours Mondays 12 to 1 and Fridays 2:30 to 3:30 p.m. And these will always be held over in Valley Life Sciences Building 2084, okay, where the GSIs hold their office hours as well. So you can find me there at those times. And like I said, I encourage you to come with your questions and we can talk about the material that we're covering in class. And what I will do for you guys is basically a week ahead, I'm going to be posting the slides for class on DSpace. So you can go to DSpace ahead of time and print out the lecture notes if you would like to do that. You can see the material that I'm going to be covering in class, and then you can look at the book and see what the chapters are covering that are related to the lectures. All right, and so for today, we're basically covering some of the material that's in the introductory chapters of the Campbell book. So if you look in the Campbell book, you'll see that the first chapter, of course, is just kind of what is biology, and then the next three chapters, chapters 2 through 4, really talk about the chemistry that underlies biology. It's very basic chemistry, just some things that probably for most of you are going to be review, things that you might have learned in high school or in an introductory chemistry class. And I encourage you just to quickly read through that material. Not so quickly if it's new to you, you need to really understand it, but for most of you, it probably will be review. And we'll talk about some of the things today that I think you want to really make sure that you understand, so that in future lectures, when we're talking about big molecules like proteins, DNA, RNA, and lipids, you'll understand the chemistry that underlies the behavior of those types of molecules. All right, so be sure that you do go through that material and just make sure that you're familiar with it. All right, so today we're going to be talking a little bit about the chemistry of life, properties of water that are essential in biology, and some of the things, kind of the themes that I think you'll see running through Bio 1A, the material that we're covering, that are very interesting and very important and kind of underlie our thinking about all of the aspects of biology that we're going to be covering in this class. And probably the most profound or most important theme in Bio 1A is, for me at least, is really this idea of evolution. Okay, so the idea that there's a process of change that has transformed life on Earth, right? And so what we see in biology today is evidence pretty much everywhere that life is all has evolved from a common origin. Okay, a common origin. And biology is basically the scientific study of life. So as biologists, what we do is we try to ask various kinds of questions about the way that life works. And of course, biology is a vast field, all the way from understanding the molecules of life, which is the kind of biology that I do in my own research, asking questions like how does a DNA molecule get made? How does it encode genetic information? What happens when mutations occur in the DNA molecule? What changes does that cause in the cell? How does that cause cancer? Things like that. Okay, questions like that. We can also ask questions at sort of a higher level. We can ask, how does a single cell develop into an entire organism? How do cells become specialized to do certain kinds of jobs? And again, the basis of that really in the end is chemistry, right? Chemistry of those cells. We can ask things like, how does the brain work? Right? That's a really important and interesting sort of aspect of biology, neurobiology. How does the brain work? And again, lots of chemical principles there. And then we can ask things like, how do living things interact in communities? How do bacteria communicate with each other? How do they grow in certain kinds of environments? How do they grow in our gut? Right? What are they doing there? There's lots and lots of interest in this field right now in biology. So, many different kinds of questions that we can ask. And biology, of course, is an experimental science. So we're always trying to think about how can we pose a question in biology that we can actually answer experimentally. How can we design an experiment to try to figure something out and do it in a way that's controlled so that we can interpret the data and then understand and try to move forward to the next question that we'd like to ask. So, one of the things about the book for me, and really for any textbook, is that when you read the book, there's a lot of facts in there, and you might feel like, this is kind of dry, right? It's kind of boring. All these facts are here, and it doesn't feel very alive. But I want to assure you that biology is very much alive in the sense that it's a very actively researched area. There's many people working on different aspects of biology, and actually our ideas and our understanding of biology change over time. Things that we thought we once knew, sometimes they change as we do more experiments. We get more data, we get more evidence. And so that's something just to keep in mind. And as you're reading things in the book, you can try to ask yourself, I wonder, how do we figure that out? And try to think about, how would I try to ask that question if I were trying to do an experiment to figure out the answer? Right? So that's kind of a fun way to approach it, so that it's more like a set of puzzles, a set of problems that you're trying to understand, rather than just a dry set of facts. And as we go through lecture, I'll try to give you guys some examples of things that we're doing in our own lab, in my own research here at Cal, things that other people are working on, things that you might be reading about in the popular media that speak to different kinds of experiments that people are doing in biology, so that hopefully you'll get a little bit better sense of how biology is really connected to many of the things that we do and think about in our daily lives. Okay, so as I said, evolution kind of underlies everything that we do, and so biology is an experimental science. I'm going to do an experiment here. Okay, I'm going to try to show you a video. So let's see if we can get this to work.
If every living creature on Earth descended from one species that could perform life's basic functions, replication, metabolism, etc., then not only should we inherit those functional capabilities, but we should also inherit the structures used to perform these functions. So a testable prediction of the idea of common descent is that all life should have similar structures that execute life's basic processes. And they do, down past the cellular level and down to the molecules that support life's processes. All life on Earth shares the same molecules that allow life to function. Regardless of species, the polynucleotides, like DNA and RNA, polypeptides, like proteins, and polysaccharides, like starches and glucose, are identical. DNA, RNA, and proteins all have the same chemical form in spite of the fact that there are dozens of possibilities that would work. All life uses the same four molecules: adenine, cytosine, thymine, guanine, in the DNA ladder, although there are more than 100 that can be used. All life bases its replication on the duplication of the DNA molecule. The proteins found in all life on Earth use the same 20 amino acids in their makeup, while there are almost 400 that could have been used. All life on Earth shares the same universal genetic code built into its DNA. The letters on the DNA ladder, taken 3 at a time, form coded instructions as to which amino acids should be joined together to form a protein. Every species on Earth uses the identical code to perform this function. Bacteria uses exactly the same code for making proteins that humans do. All life on Earth shares the same metabolic pathways. In all life based on cells with a nucleus, from amoebas to blue whales, glucose is metabolized in the same 10 steps, in the same order, using the same 10 enzymes. Thousands of new species are discovered yearly and have their DNA proteins sequenced examined. Nearly 50 million new bases are sequenced every day, and everyone is tested of the theory of common descent. It passes every test. There are enough different possible genetic codes, all functionally equivalent, and all using the same amino acids. For every species that has ever lived, it has its own unique code. If there were no common ancestor from whom all life inherited this code, it would make sense to expect a wide variety of codes. This would protect each species from interspecies viral infections. The lack of variety indicates common origin.
Okay. So that probably wasn't too surprising to most people here, but I think it's very important to think about evolution as we go through this material. And one of the things that they point out in that little video is the fact that, and this is very true, this is again, this is an experiment that's been done many times in many different systems, is that it's often possible to replace one protein, for example, from a particular organism with a protein with a similar function from another organism and have it work. So in other words, you can make a mutation in a yeast cell that knocks out a critical enzyme important for sugar metabolism, and without that enzyme, those yeasts are unable to grow, okay? And then you can replace it with the enzyme that has the similar function from, say, a human cell. And you can show that when you do that, the yeasts are now able to grow again. They're able to metabolize sugar again. Okay, and so again, this is just one of the themes that we find in biology that I think is extremely interesting, and it really underscores the molecular evolution of all life, the fact that all organisms are related to each other, they use the same genetic code, in many cases they encode the same kinds of proteins that have the same kinds of functions. Okay, and so that's something that you just want to keep in mind as you're thinking about the macromolecules that we're going to be talking about in my section of the class and how they behave. Because a lot of the, so if you think about research in biology, a lot of the original work and the way that we know a lot of the things that you'll read about in the book and that we'll talk about in class actually come from model organisms, things like experiments that have been done in bacteria, in yeast, in other kinds of fungi, organisms that are fairly easy to manipulate genetically and that we can study in the laboratory quite easily compared to humans, for example. But a lot of the principles that we learn from those model organisms turn out to reflect the same kinds of behaviors of those molecules in human cells. So a great example of this is a friend of mine doing research at the National Institutes of Health, studying how bacteria deal with mutations that occur in their DNA. How do they repair mutations that happen due to radioactive damage or other kinds of environmental stresses? Her name is Wei Yang. And what Wei discovered when she was doing her research was she discovered a molecule called MutS that is an enzyme essential to the repair of DNA in bacteria. So she was studying this process. She was looking at the molecular structure of this enzyme. And what turned out to be, what sounded kind of initially like it might be sort of an obscure line of research studying how bacteria fix their DNA, later turned out to be the case that MutS has a matching protein, a very similar protein, a very similar function in human cells that is the most frequently mutated protein in human colon cancer. Okay? And so all of the work that she had done in bacteria to study the function and structure of MutS and the way it works turned out to be very, very relevant to understanding how MutS, the homolog of MutS in human cells goes awry in human colon cancer and how one might deal with that therapeutically. So that's just one example where studying a system in a model organism turns out to be very relevant to human health. And like I said, there's lots of cool examples like that. Okay, so as I mentioned, so please look through chapters 2 and 4, the chemical foundations of life. I think all of you know that biology is multidisciplinary, meaning that we have to know at least something about chemistry and physics to be able to understand biology, and increasingly mathematics. So mathematics is a field that also is very, very relevant to a lot of the kinds of research that's going on right now in biology. We know that living organisms are subject to these basic laws, and so you need to know basic definitions of matter and the chemistry of water and the chemistry of carbon so that you can think about the material that we're going to be talking about and understand the things that we're going to be discussing in class. All right, so you know that organisms are composed of matter, and matter is basically anything that takes up space and has mass. And what's matter made up of? Well, it's made up of the elements, right? These guys over here. And again, probably most of you know this, remember this from high school chemistry or introductory chemistry classes, that an element is a substance that can't be broken down into any other substances by chemical reactions, okay? And so that's an element. A compound is a substance that consists of two or more elements in a fixed ratio, okay? That's a compound. A compound has characteristics that are different from those of its component elements, all right? So oxygen, is that an element or a compound? Element, yeah. What about iron? Also an element, yeah. Water and carbon dioxide, compounds, right? Okay, exactly. So, and the thing is, so oxygen is a molecule, right? It has two atoms of oxygen that combine to form a molecule of oxygen, but it's still an element, right? It's not a compound. Okay, and then this is just illustrating something that you all know certainly by experience, and that is that when two elements come together to form a compound, the properties of the compound are very different from the component elements. So this is just an example for table salt, sodium chloride, and these are the component elements, sodium and chlorine, neither of which you would want to consume, certainly. But when they come together to form a salt, sodium chloride, that is a compound that we consume every day. In fact, we need to have in our diets, and many organisms do, right? And so that's an example of a compound. And in the book, they talk about things like, again, things that are probably familiar to most of you. What is a covalent bond, right? What is an ionic bond? So sodium chloride is an example of a compound that comes together to form ionic bonds, right? By ionic bonds. So you need to just make sure that you remember a lot of that basic chemistry from your background. Okay, so there's 92 natural elements that are, and of these, about 25, roughly a quarter, are essential to life. There's 4 of these that make up really the vast majority of molecules that we find in biology, and that's carbon, hydrogen, oxygen, and nitrogen, right? 96% of living matter is made up of those 4 elements, compounds made from those elements. And so when we talk about proteins, when we talk about lipids, carbohydrates, DNA, and RNA, the nucleic acids, you'll see these 4 elements over and over again as components of these big macromolecules. So it's important to just, one of the chapters, I think it's chapter 4, reviews the chemistry of carbon.